Welcome everyone. Today I will be showing you how to perform a simple fixed backbone design using InteractiveROSETTA. This process is actually fairly simple, so this tutorial should help you get up and running with designing proteins quickly. The first thing we are going to need is a protein to redesign, so let's take our favorite protein to use for these demonstrations. Now let's change the protocol to Protein Design Fix BB. The Fix BB panel looks like this. There are a lot of buttons but they are all quite simple to use. First we have to select some residues that we would like to redesign in either the sequence window or in PyMall. Let's take the first 5 residues for this example. If I click the Add button, you will see that all of these residues have been added to the grid down here. This column indicates what amino acids will be available from which Rosetta will choose when designing the protein. It defaults to the wild-type amino acid. Why is that? Look at all these letters up here. If I click on the buttons, they toggle amino acids on and off. Since I did not have any amino acids turned on, the selections default to the wild-type residue. Let's turn on a couple of residues and then select the first 3 residues. When I click Add, these positions get the subset of amino acids turned on with the controls. If I select new residues and add them, they also get the subset. This X button will turn all amino acids on or turn all amino acids off, so it's useful if you do not want to spend a lot of time clicking letters. This grid is clickable. When I click a residue in the grid, PyMall will do a neighborhood view of the selected residue, so I can easily see what the current neighborhood of the residue is while determining amino acid selections. Once I have selected a row in this grid, I can delete it by pressing the Remove button. Restrict will remove everything from the grid except my current selection, so if I select a new subset of residues and hit Restrict, only the selection will be in the grid. Clear obviously gets rid of everything in the grid. All will add all residues to the grid. I should note that if you have multiple models open, you can only design one at a time, so pressing All will take all residues from the first selected model, or the first model if nothing is selected. Okay. Let's generate a somewhat real design palette. In Rosetta terminology, this is called a res file. So it can take a while to generate these setups, so there is a Save button down here where I can save all of the information in this grid to a Rosetta res file. If I click the Save button, it will ask for a file name, and then the res file will be generated. This is a valid res file, so it should be compatible with other Rosetta protocols. If you are familiar with running Rosetta at the command line, you can use this feature to facilitate generating res files if you wish. The Load button will load the data saved in a res file. If you attempt to load a res file that was generated for a model different than the one currently loaded, the data will attempt to be loaded but invalid design fields will be discarded, so keep that in mind. There is one toggle button over here that can influence design. What does this mean? This specifies how all residues not included in the above grid will behave during protein design. If net row is selected, then all the other residues will not be touched at all during design. If net AA is selected, then all the other residues will have their rotamers changed during design, but the wild-type amino acid will be preserved. Performing design in net AA mode will be slower because all residues are mutable, so keep that in mind. If you are only designing part of a protein, it is not necessary to redesign everything, so if you want the residues in the neighborhood to have their rotamers repacked, add them to the grid but default to their wild-type amino acids. Okay, when you are ready, just click on the Design button. This process will go fairly quickly because the design was a simple one. More complex designs will take longer to complete, but usually they still finish relatively quickly. Okay, the design is done already. Let's take a new look at the grid. The labels have been updated over here. You can see that they represent standard mutation notation indicating what the original amino acid was and the new amino acid selected by Rosetta. There is also a color coding going on in the grid itself. These colors qualitatively represent Rosetta scores. The more red the color is, the worse the scores, and the more blue the color, the better the score. If I click on the rows, I will again get the neighborhood view of the residue, but there is even more information now. You can see that the coloring applies in the PyMall view as well. There are also some green structures. The green structures are the original protein, so you can easily compare the structural environments before and after. The number represents the total Rosetta score on this residue. You will notice that this drop-down menu is unlocked after finishing a design. His menu contains all of the Rosetta scoring terms. So I can select one of these options and the colors both in the grid and in PyMol will be updated to qualitatively reflect only the indicated scoring term. So if I have a residue with a bad total score, I can flip through the scoring terms in this menu to identify which terms specifically are giving rise to the bad score. We are not done yet though. Lastly, I have to click this Finalize button to either accept or reject the changes made by Rosetta. If and only if I accept the model will the structure be updated. As you can see that the sequence was updated after I accepted the design. Anyway, that is all there is to fixed backbone design in Interactive Rosetta. See you in the next video.